Thank you for giving me the opportunity to present our research at the International Horticultural Congress. My name is Jeonjung Kim, and I am a PhD student at Cheonam National University. Today, I will present our study on the fine mapping and candidate gene identification of MS2, a novel restorer of fertility locus in onion. The presentation is organized as follows. Our group mainly studies onion. As shown by these indicators, onion is one of the most economically important vegetable crops worldwide, and its importance continues to grow. Onion cultivars can be broadly classified into open-pollinated varieties and F1 hybrids. Currently, F1 hybrids are widely used in commercial onion production to exploit heterosis. For F1 hybrid seed production, preventing self-pollination in the maternal parent is essential. In onion, this is achieved using cytoplasmic male sterility, or CMS. CMS is a trait in which plants fail to produce functional pollen. It is referred to as cytoplasmic male sterility because the genetic factor responsible for male sterility is located in the mitochondrial genome. Male fertility in CMS plants can be restored by nuclear restorer of fertility, or RF genes. In onion, F1 hybrid breeding based on CMS uses a three-line system. This system requires three different lines, a male sterile line, a maintainer line, and a restorer line. These lines are defined by their cytoplasm type and RF genotype. Rapid and accurate identification of these lines is essential for the successful use of this system. Molecular markers are useful tools for this purpose. However, to develop reliable molecular markers, the causal genes first be identified. Through extensive research, our group identified ORF725 and ORF219 as CMS-inducing genes in onion. We also identified ACP-MS1 as a candidate gene for MS, an RF locus. Recently, our group identified MS2, a novel RF locus in onion. Using BSR-seq, we developed 20 molecular markers linked to MS2. Furthermore, unlike MS, we found that MS2 restores male fertility by reducing the expression of ORF725, a CMS-inducing gene. This finding suggested that MS2 may encode a pentatricopeptide repeat, or PPR, protein. However, the candidate gene underlying MS2 was still unknown. If the dominant MS2 allele is introduced into a breeding program, the current marker system cannot accurately predict the male fertility phenotype. Therefore, we needed to identify the gene underlying MS2 and develop a reliable marker for this locus. Recent advances in sequencing technology have enabled the assembly of high-quality genomes, even for crops such as onion, which has a large and complex genome. In 2023, a chromosome-level onion genome assembly representing approximately 99% of the genome size was published. Therefore, we used the high-quality whole genome sequence to identify the candidate gene underlying MS2. During this analysis, we also found that MS2 partially restored male fertility in a specific CMS cytoplasmic background. First, I will talk about the identification of the candidate gene underlying MS2. Using the published onion genome, we physically mapped the 20 MS2 linked markers developed in our previous study. Perfect collinearity was observed between the linkage map and the physical map. Therefore, the genomic region harboring MS2 was narrowed down to a 12.5 megabase interval. However, this interval was still too large, so further narrowing was required. We used recombinants and additional molecular markers to further narrow this region. First, we aligned the BSR-seq reads generated in our previous study to the 12.5 megabase candidate region, and developed 11 new molecular markers. We then performed fine mapping using these markers and the 12 recombinants identified in the previous study. As a result, the 5' boundary was defined by one recombinant at IND 4.45 marker, and the 3' boundary was defined by another recombinant at H7.64 marker. Finally, the MS2 candidate region was narrowed down to a 3.19 megabase interval. Gene annotation within this interval identified 15 genes. Interestingly, among these genes, we identified a gene predicted to encode a mitochondrion-targeted PPR protein comprising 876 amino acids. We designated this gene ACPPR876. Next, we carried out several analyses to obtain further evidence supporting ACPPR876 as the gene underlying MS2. First, we searched paralogs of ACPPR876 in the onion genome using its protein sequence as the query. We identified 4 closely related paralogs, which formed a gene cluster with ACPPR876 near one end of chromosome 2. Next, we analyzed their phylogenetic relationships with previously reported RFPPR proteins from other plant species. All 5 proteins clustered with known RFPPR proteins and were classified as P-type PPR proteins. Then, to identify sequence polymorphisms in ACPPR876, we obtained the full-length genomic sequences of the gene, including the 5' and 3' flanking regions, from male fertile and sterile plants. We identified 10 SNPs in the coding region, including 4 non-synonymous SNPs. Of these, 3 resulted in amino acid substitutions within PPR motifs. Notably, one substitution changed the 5th residue of the 4th PPR motif, one of the key positions constituting the PPR code. This change in the PPR code may alter target RNA recognition. Most variants were concentrated in the 5' region of the gene. This pattern was likely generated by intragenic recombination. Previous studies have suggested that the clustering of RF-like PPR genes can promote this type of sequence variation. Among the variants in the 5' region, we identified a 446 base pair insertion in the putative promoter region of the fertile allele. Because this indel was expected to affect ACPPR876 expression, we examined the expression levels of ACPPR876. Both RNA-seq and RT-qPCR analyses showed that overall expression of ACPPR876 was low, but its expression was higher in fertile plants than in sterile plants. In addition, none of the other genes in the delimited region were differentially expressed between the male fertile and sterile bulks. These results suggested that the 446 base pair insertion may enhance ACPPR876 expression and that the PPR code change may alter target RNA recognition. Collectively, these variations may contribute to the fertility-restoring function of ACPPR876. Based on this insertion, we developed a molecular marker, IND876. This marker clearly distinguished the MS2 genotypes. When we analyzed 128 recombinants from large segregating populations, IND876 genotypes perfectly co-segregated with the male fertility phenotypes. Using this marker, we analyzed 250 Korean breeding lines and 108 exotic accessions. None of the Korean breeding lines carried the dominant MS2 allele, whereas it was detected in 29 exotic accessions. Finally, we found that the stability of MS2-mediated fertility restoration depended on the cytoplasmic background. In onions with CMSS cytoplasm, the dominant MS2 allele partially restored male fertility. Pollen viability assays and electron microscopy revealed that many pollen grains were non-viable. Notably, partial male fertility was also observed in some CMSS plants carrying dominant alleles at both the MS and MS2 loci. This pattern was consistently observed over 5 years. To evaluate the effect of partial fertility restoration on seed production, we conducted self-pollination tests. Compared with CMSR plants, plants with CMSS cytoplasm and the dominant MS2 allele showed a significantly lower seed set, reflecting the incomplete restoration of male fertility. However, the seeds produced by these plants germinated normally. These results indicate that partially restored plants can still produce viable seeds, although their seed set is reduced. In conclusion, we identified ACPPR876 as a strong candidate gene underlying MS2, and confirmed the presence of the dominant MS2 allele in diverse accessions. Previous studies have shown that CMSS is the most widely used cytoplasm in onion F1 hybrid breeding. However, our results showed that MS2 partially restored male fertility in onions with CMSS cytoplasm. Because stable control of male fertility is essential for F1 hybrid seed production, the dominant MS2 allele may be undesirable in F1 hybrid breeding programs. In this case, the IND876 marker can be used to identify and remove plants carrying the dominant MS2 allele from breeding materials. This marker-assisted selection will contribute to more efficient F1 hybrid breeding in onion. That concludes my presentation. The work presented today has been published in 2 papers. I would like to express my sincere gratitude to Professor Sung-Gil Kim for his guidance, to the funding agencies that supported this research, and to all members of the Horticultural Crop Genetics and Breeding Laboratory at Chatham National University for their support. Thank you for your attention.